My name is Molly McDonald, and this is my case study 2 about azithromycin, or also known as AZT. So, a quick overview of AZT, also known as azithromycin, or more commonly by its trade name, Retrover. It is an antiretroviral therapeutic agent that is used to treat HIV and AIDS. It functions by selectively inhibiting HIV's reverse transcriptase, which inhibits the virus's ability to infect a cell with its genetic material by acting as a chain elongation blocker. This blocks many this blocks any viral reproduction and acts as an antiviral drug. In order for AZT to properly work, it has to be converted by host enzymes to AZT triphosphate, which is also known as AZTTP. Research shows that AZT can cause changes to the mitochondrial structure and the respiratory chain enzyme activity, which overall will reduce ATP levels. So, the process of AZT being turned into a usable form. So, AZT is a thiamide analog that is converted into an active triphosphate metabolite, which is also known as AZTTP, as previously mentioned, and this occurs through the sequential phosphorylation of azidomythine. And this essentially is when amino acids of a protein are phosphorylated in a specific order, which ensures the phosphorylation of a certain residue is required for the next step. Protein kinases facilitate this process and work to add phosphate groups to proteins. This process is essential for protein function and the regulation of enzymes, channels, and intracellular signaling molecular activity. So the mechanism begins with AZT, and AZT becomes monophosphate through thymidine kincell being involved. The AZT monophosphate becomes AZT diphosphate with the enzyme thymidylate kinase being involved. And then AZT diphosphate becomes AZT triphosphate, which is converted into its final active form. And this happens with the help of the enzyme nucleoside diphosphate kinase. And this entire mechanism occurs in lymphocyte cells. So, a quick overview about HIV and AIDS, which are the topic of the disease that we are talking about. HIV, which is also known as human immunodeficiency virus, is a virus that attacks the body's immune system, and this may lead to AIDS if left untreated. AIDS is also known as acquired immunodeficiency syndrome. There is no cure made yet for HIV, but there are proper medical care that helps control the virus. Symptoms of this virus include a fever, sore throat, swollen lymph nodes, rashes, muscle aches, night sweats, mouth ulcers, chills, and/or fatigue. It is spread through certain body fluids such as blood, semen, rectal or vaginal fluids, and breast milk, and this is because it has previously come into contact with a mucous membrane or damaged tissue. HIV treatment works to reduce the amount of HIV in the blood and makes the amount, which is also known as the viral load, low enough that tests cannot detect it and you are no longer completely contagious. Um, the process the process of how AZT can knock down HIV and/or AIDS. So AZT is a reverse transcriptase inhibitor that works to fight against retroviral infections. And this works to revive the previous immunity of the patient. Reverse transcription in HIV entails a biochemical process that functions to polymerize DNA over an RNA template, and the enzyme reverse transcriptase is involved in this. And in contrast, transcription is a biochemical process that functions to polymerize RNA over a DNA template and involves the enzyme reverse transcriptase. Both of these mechanisms use DNTPs, which are also known as deoxynucleoside triphosphates, and these work to lengthen the DNA or RNA chain of the process occurring. These DNTPs incorporate hydroxyl groups into the reaction that form phosphor phosphodiester bonds that link nucleotides to lengthen the nucleotide chain. If a hydroxyl group is not incorporated into the reaction, then the mechanism will come to a pause because the DNA or RNA chain will be unable to become longer and the viral infection will therefore be dissolved. So, in conclusion, overall, AZT works to pause chain elongation during reverse transcription in HIV, and this works to halt any hydroxyl groups from joining the reaction on the ribose sugar of the given nucleotide. When AZTTP stops chain elongation from occurring, The viral genome is paused and becomes unstable, ultimately destroying itself and diminishing the amount of HIV from the blood. Here are all my research citations, and thank you so much for listening.